We have been working several years. When I used to, to work in university, and now a little bit we moved forward. So, in our own experience and missing link for today, especially pharmaceutical industry, is that we always, as previous speaker is excellent talk and all of topic that we cover, but it's come to our attention that is big missing links. Is like, even, to one of previous speaker is mentioned that is, it is how we should avoid, money, how we should avoid wasting time. But when we come to final clinical trial, stage 3 or 4, as you know, you read, and so you say, okay, failed. Why? And so for this reason, so based on our experience, I'm going to talk a little bit about is key component of cellular compartment called mitochondria. I don't want to go into detail about mitochondria, you know better than we know, because it is only one organelle contain own DNA. So autonomic system. And unfortunately, is missing links, and we are going to to little bit take into account within one of devastating disease, neurodegeneration, Alzheimer case, and other one is, another devastating cancer, what's kind of relationship, and some of clinical results is based on our own experience. And so I wish to take. So this is, our facility, and before to go to talk, I wish to thank you, all of these people. So you can see from almost all over the place in United States as well as Europe, and part of, to some of Middle East, to university, all over there. So I'm very pleased, to thank you, all of this contributor. Without their help, we will be, this big, I'll say, ongoing multi-layer project impossible. As you know, when we're young, we don't, we feel good and we don't have any disease and everybody fine and, life is going well. But it's by age you get all kind of problem. Like, to is car, what type of car good? Best car is new car. So how we can deal with aging? Because aging, what's mean disease? Disease is accelerated aging, we know. And aging requires time manner, and metabolic structural change, and why we measure all the biochemistry, all of laboratory tests. And this is first effect, what type of cells, highly metabolic, like, what we get. Say, okay, that is cardiovascular disease or, neurodegeneration, so neuron effect, so highly metabolic, to disease. Aging, to this multi-layer process, and here main factor is again missing link is that we always say, okay, let's say the endocrinologist, I deal with the glucose level, I don't know what's going on with cardiovascular system, I don't want to know what's memory function. This is actually just a misinterpretation why you will see is one of challenge in Alzheimer's disease. Until today, any pharmaceutical industry able to bring on table anything else, or in cancer as well as we kill. Why? How we can deal with by using maybe mitochondrial antioxidant metabolite, because by ages this is decreased and already shown this can be preventive and against oxidative stress was effect by age. You can see with the tiny organelle. I can cover for 20 or 25 minutes. Well, I'm going to just to concentrate one of two components. It's called lipoic acid and acetyl-L-carnitine. So if you need, to full length of lecture, I'll be more than happy. You can see my email in program, and I'll be more than happy to share with you. But we need to get, you have to have permission if you want to even use for your need. By age animals, we treat it with acetyl-L-carnitine lipoic acids, and you can see that is neuron in compared to aged group can be improved structurally. This is picture taken by using electron microscope. Quantitative study also show that is treatment, especially combinative treatment, effect more visible in old animal. This is another assay we developed, so you can see that processes occur every organ or tissue. You can see liver and brain. MDA level is, by age is increased significantly. So again, go back to mitochondria. Different type of damage of mitochondria are increased by age and can be diminished by using selective antioxidant. As you know, mitochondrial membrane potential, cardiolipin, as an indicator of aging, by using fluorescence per cells, you can see with this also cardiolipin level is decreased by age, and fluorescence positive or dead cells are increased by age. And with the Morris water maze testing memory, you can see that is only combinative treatment able to give us much better and significant improvement in memory function. Moreover, as you know, heavy metal appears to be one of, I'll say, silent killer. You can see iron and copper level is increased by age, but can be diminished by using combinative mitochondrial antioxidant. With adjacent section, marker for oxidative damage, you can see that much better diminution, able to see one is combination, treatment. Moreover, with this oxidative stress dependent, staining by using monoclonal antibody oxide 8-dehydroxyguanosine, and this is mostly cytoplasmic, and you can see one is treatment, one DNase you cannot, so is no any abolition you can see. No pre-treatment by RNA, so it's shown that is with oxidative stress or RNA dependent. Anti-lipoic acid, also adjacent section is qualitative serial section show that is improvement. Mitochondrial complex 2, mitochondrial complex 4, mitochondrial complex 5. So this is show that improvement, it is systemic. So based on this, our conclusion is that free radical of aging, this is to appear to be energy misbalance, and especially in brain, and most likely that's another human disorders is caused by pro-oxidant and antioxidant, which is making oxidative stress with outcome cell damage and organ function. And so with the first time we're able to show systemic analysis, I can show you here all of us with immunology, all of that we published, and our collaborator published several, and so you can search on Medline. Go back to human disease. So Alzheimer's disease, as you know, I don't want to go to what's mean Alzheimer is, to memory dysfunction by age. You can see that is Medicare in patient database is most of case who is Alzheimer's patients. So leading hypothesis is that in our understanding is you can see compare normal and Alzheimer brain, you can see but this is age associated disease. So strictly age dependent. Different theory that can be involved in progression as an incident, probably antioxidants, which is down by age, is most likely protective. So I'm going to just concentrate a little bit oxidative stress. So this is our work in theory, 2008, we were able to show that is, current golden theory called amyloid has nothing to do with Alzheimer. We found that this is, biopsy from Alzheimer patients show control versus, damage. You can see is mitochondrial damage appear to be primary before any kind of amyloid deposition. This is quantitative study. So moreover, we developed techniques called mitochondrial DNA visualization by using indirect colloidal gold. This is one of highly cited paper. And this is also related to mitochondrial enzyme. You can see this is the dot in the internal side of internal membrane. As you know, mitochondria on the one organelle that says double membrane. It shows positive, to DNA containing signal in Alzheimer brain. Moreover, nucleic acids damage also shows positive with that gold particle correspond to 8-hydroxyguanosine. Moreover, with no count staining tissue show that heavy metal also appear to be. So we found that is based on I just here mentioned a few of our recent publication is that mitochondria is a primary target for development Alzheimer's disease can be, in different type of model we showed that this can be as a, new strategy for against for disease. It is true, we have to see with this transgenic animal or any other model. You can see same type of mitochondrial lesion in vascular cells on brain. One, as a reviewer of many journal, I'm sure all you guys also doing, very often when you see papers say, oh, its control show no change and almost zero. It's not true. Aging himself induced lesion. But question is, if this lesion is not significant, and therefore is, you don't see any, organ function failure, or if you see, it's very minimal. But go back to transgenic animal, you can see without any amyloid deposition, perikaryon area or perinuclear region where there's a high metabolic activity show mitochondrial over transform into vacuolar structure. It is a quantitative study, also correlated with human study. Moreover, amyloid precursor protein containing positive gold particle, so protein overexpression occur before any type of lesion or can be. Meantime, chimeric or mutation of mitochondrial DNA by age, you can see only few positive signals. But go back to Alzheimer mice, you can see is cluster type of positive. Moreover, not only neuron, but other compartments such as vascular system in brain, microcirculatory system also show positive DNA signal in atherosclerotic type of vessels, including, to chimeric probe also show cluster type of positive signal. So we show that mitochondria, vascular endothelium, and neuron appear to be key component during the development of Alzheimer's pathology, transgenic animal. This is, to reference for this project. So it's true, we have to get positive effect. We show in aged animal, but maybe we can get same positive in transgenic animal. We use this model because this is as APOE4 is relevant to late onset form of Alzheimer's disease. Is same techniques, I don't want to go into detail, we applied here. You can see blood flow is changed by age and is decreased by regional blood flow in transgenic animal, especially in different brain region. Cortical blood flow in wild type of transgenic animals show APOE mice or transgenic animals show decreased blood flow. You see, age himself, perivascular control group does not show or show very minimal change in very late onset, so age. This is almost correspond to about 8 years old human. So, but you can see even young animals show is stress reaction lesion in transgenic. Want to get old is pathologists much more, and including vascular previously here in neuron, and also we're able to show by using memory testing that is treatment even transgenic animal. We know this animal has lacking for gene show positive to improvement, and therefore we're able to show is change in parameter that correspond almost to human Alzheimer, and moreover, implication of mitochondrial antioxidant can be diminished. And this is first time it was shown in large scale of paper and work, also from clinical study. Also, I want to show a little bit. So, if it's work, if our theory correct, work in age group, it's work in transgenic animals and overlap with human Alzheimer. Maybe we can try to treat people. So in a very simplified way. So we added same pattern of, to medication for the people that's, to in this group. So this is demographic clinical profile of patients. It's different, you can see, it's different group. And moreover, we should take into account, I didn't get a chance to ask question previous speaker, what's mean, to coexisting condition, because just only diabetes not only has, a higher blood glucose level, but also liver failure, creatinine level, all of that. How we should take into account. So therefore, as you can see, aged population has large scale of coexisting disease. So we say I'm going to focus on diabetes, doesn't work. So you see, we're in with a small group of presentation, or 156 total patients, and after 60 months. So you can see during this time, we tested them time to time. You can see near a behavioral cognitive state especially after 24 months, we get, to very stable, to improvement. And this is also maybe I just work, just get published today. I just received recently email. So Alzheimer's type this paper is just get, you can see that is multi-level, to complication induced with, in genesis of dementia or coexisting metabolic disease condition. Maybe diet in covering with other component should be taken into account, especially, treating elder patient. So we showed that implication of mitochondrial antioxidant, not single, but combination with different type of coexisting, we should take into account patient's condition, and we are able to gain significant improvement, cognitive empowerment after 5 years. This guy, you can see, is already old. We get very excellent, stable. They not only get down, but they also get, improvement. So this is reference for this part of, project. You can see that we are able to, several paper in clinical aspect. So it looks like in dementia or Alzheimer case or brain-related disorders, mitochondria appear to be primary. Does this work in case of, cancer? And as you know, is free radical or nitric oxide is, one of label molecule, can go through everywhere. So this is, we hypothesize maybe, kind of links between this. So this is electron microscopy immunocytochemistry obtained from patients undergo surgery for hepatocellular carcinoma. You can see that show highly toxic condition, which is also lesion and disorganization tissue, large scale of damage mitochondria or compensatory form called this also coexist inducible form of nitric oxide, positive indicating highly toxic or hypoxic condition. Moreover, in this condition, we see, almost absence of positive reaction for normal physiological marker for nitric oxide, endothelial specific nitric oxide synthase. This is colloidal gold triple label. This technique is also developed by us. We found what is, tumor condition characterized overexpression of all of three isoform as well as endothelin 1. Which was a constrictor and as well as is a pro-tumor precursor, is seen in tumor tissue. But when you diminish, you can see his reaction is very specific. Go back to brain, one of deadly form of brain tumor is glioblastoma. You see microvessels as well as tumor tissue. You'll see giant type of mitochondrial lesion or fusion or diffusion appear to be, part of his tumor growth. It is, but you see, if you distance from tumor, you see less lesion. And meantime, you can see central region of tumor tissue, not only, to vessels, but other tissue or neuron, as well as, to astrocytes show cluster of mitochondria-derived lysosomal structure. You see this control and intermediate area show is oligodendroglioma case, another form of tumor, show minimal or almost, to very lesion, so minimal lesion. You can see how tumor cells go to throat, and vessel barrier. But you see, it's characterized destruction of mitochondria, which appear to be primary, to marker in this case. We did, in situ hybridization. You can see, go back to it's animal model of transgenic, as well as, the case of human Alzheimer and, human tumor. All has same pattern. So therefore, is specificity of the tissue, non-specific of the reaction. You can see it's also in the vessel. Show mitochondrial power containing mutant, DNA signal, and are located specifically mitochondria-derived lysosomal structure. So we believe that metastatic colorectal cancer as well as malignant cancer are character or expression of nitric oxide synthase, immunoreactivity, and this is most likely induce mitochondrial damage in all of cellular compartment. Moreover, this is elevated, coexist with elevated level of endothelin-1 before cancer growing tumor and destruction of a tissue occur. If this also is relevant reference for this topic, just come out. And what we can do? Most likely, free radical induced DNA damage, which are is case of Alzheimer's, a different type of model, also we're able to show, as well as human tumor, making us kind of, direction, but most likely, if we target mitochondria, we're able to, to gain a very positive, to possibility, targeting in cancer and almost, eliminating. And yes, I wish the special thanks, financial support, NIH, Alzheimer, Philip Morris, American Cancer, as well as British Heart Foundation as my long-lasting collaboration, and our institution for this. And I'm specifically wish to thanks to my manager, which is an excellent, hard-working guy, almost 24 hours, and also my daughter. She's helping me time to time, and she's just graduated from university. And this is, recent book just come out. It's about, what we should take into account based on our past experience and ongoing work in the future that can be, to implicate it in human disease. And this topic also very, highly, highlighted in, TV as well as some in the media. And I wish to, and thank you for your time and listening.